So my talk will be essentially a talk about analysis of Luminex data. So as you may or may not know, Luminex is a kind of multiplex version of ELISA. It allows the measurement, and I'll go into some details about that. It allows the measurement of multiple analytes simultaneously, and as such has been quite widely been adopted in many labs across the world. But at the same time, it's known to generate data that is quite noisy as well. So this project was about taking a post-hoc approach after the data is being generated and taking a second look at the data to see how we could improve the signal-to-noise ratio of this kind of data sets. So this is a schematic of how the so-called Luminex XMAP technology works. So this technology is meant to enable simultaneous detection and quantitation of multiple secreted proteins, such as cytokines, chemokines, and growth factors. And the idea is that a target extracellular analyte is sandwiched between a capture antibody and a detection antibody. The antibodies are attached, the capture antibodies are attached to magnetic beads, such that each magnetic bead is coated with capture antibodies that are specific to the same analyte. And internally, also the bead carries a mixture of two dyes, a red and an infrared dye that are specific, the mixture is specific to that analyte. So the assay consists of a large number of these kind of magnetic beads, where different sets of beads have different specificities, and they are mixed together and run through a kind of an adapted flow cytometer, so that the bead essentially plays the role of what a cell is when it goes through a flow cytometer. And with lasers, the fluorescence is measured. There are different implementations of XMAP available. What I will show you is data that we have in our implementation at the Center for Human Immunology at the NIH, which is mostly from Biorad, but other vendors are Affymetrix, R&D, Luminex Corporation, and others. So looking at the raw data, which is ultimately what I was interested in doing, so moving away from what's the standard pipeline of analysis and seeing what else could be done by starting from scratch from the raw counts. This is a snippet of the data that is captured. So this vendor Biorad generates output files in binary format of extension RBX, but they have proprietary software that we as users have access to that allows us to export the data as XML files, and then the XML files can be parsed to extract bit level information from each well, as I'm showing there. So each row corresponds to one bead. As I say, there are many hundreds or even thousands of magnetic beads measured together in one well. Each of them, so a set of beads specific to one analyte. And the 4 columns refer to different properties. So the first one measures the forward scattering height versus area that can be used for doublet exclusion. The second column is the most interesting one because it's a fluorescence from the reporter dye. That's the one that essentially we can correlate or consider a direct measure of the concentration of the target analyte that we're interested in. And the third and fourth columns are the fluorescence from this combination of red and infrared internal dyes. And the combination of the two is designed to select one particular target analyte. So in this case, for instance, when this is an example of 21 different analytes all mixed together, and one can separate them in a bit by bit manner by looking at the combination of the two. So you see that the 21 clouds are distinct. So the analytes are grouped together into what we typically call plexes according to biological process. For instance, acute phase, metabolic, immune, and cancer are different groups of analytes, and also depending on their expected endogenous concentration. So for the Biorad technology that we have, these are the plexes we use most often. So this amounts to more than 90 analytes that can be measured. So the standard pipeline consists of a procedure of data filtering, and they end up measuring the mean fluorescence intensity for each combination of well and analyte. But then, and on top, there is a design of how the 96 well plate looks for this technology. There are two columns that are labeled standard. So those are in duplicate 2 8 serial dilutions of recombinant protein control sample of known concentration. So with those 2 sets of 8 standard measurements for which we know the concentration, we can fit a so-called standard curve of MFI, or fluorescence intensity, against concentrations, and those are the blue dots. One can fit typically a multi-parameter logistic regression curve, and with that, now the green points are MFIs of samples that have unknown concentrations, so one can translate MFIs into concentrations that way. So that standard process has good and not so good aspects. The good is that they provide absolute concentration values, and they address to some extent plex and batch effects, because we are normalizing each set of data of samples to what's contained in each plate. But the not so good is that it's based on these averages over bit distributions, these so-called MFIs, and also the fact that there is a compounded effect of error propagation, because to the error at the MFI level, we compound the errors from the fitting procedure and from the errors from the measurements in each of the standard samples. So our goal is to keep the good aspects and improve the not so good ones. The focus, well, I'll describe the focus now in a minute, but essentially run a mixed effects model of the standard curve and use it to calibrate the sample measurements. And our data is a very large, well, we've generated tons of data, but this in particular, what I use here, has many plates, which is very amenable to this kind of mixed effects model. So it's a large study on vaccination of healthy donors. So first, the first pass is a QC. Or first step is a QC of each distribution, because now I can look at the bit level distributions. I can determine what's the shape of those distributions. The idea is that for the mean or the MFI procedure, that's the standard procedure, one should have distributions that are essentially unimodal, because otherwise the mean wouldn't mean anything. So in this case, this is just synthetic data, and the way the method works for different distributions, flag the distributions as passed or failed in terms of unimodality. So we can do that on the real data that we get for each well. And once we do that and we look at the standard curves, this is for 4 different analytes, but we have this for each single analyte. We parameterize the standard curves. So the x-axis is the concentration with some spread, just so you see the different repeats. So it's 21, 42 data points for each concentration from the standard curve, and the vertical axis is the MFI that's measured. And so what we find is that a 4-parameter logistic function or sigmoidal is flexible enough to provide good fits for all the data that we need to look at. So this is the left panel is another way of looking at these same plots, and you can see for the 8 different concentration levels of the standard curves, the x-axis is just 42 replicates from 21 different plates, each plate running duplicate. And you can see that there is a lot of noise. Running a mixed effects model, really what we can do is now merge all the data coming from all the analytes, all the plates, all the batches, everything together. And the property of a mixed effect model is that it kind of borrows shared uncertainties from different variables so that one can model noise better and therefore get that cleaner data. So this works with an iterative algorithm that runs the mixed effects model once and then identifies the outliers by analysis of residuals and then fixes the outliers beyond a certain threshold to kind of smooth out the curves. And so you can see on the right how the different models, as we add more and more nuisance effects that are modeled in this way, we can get a cleaner version that still captures all the nuances and variances that are really contained in the data. So the advantage is now instead of now standardizing our data against those standard curves on the left, which are very noisy and have definitely a lot of fluctuations, now we can do it based on the data on the bottom right panel, which is smoothed out and much cleaner. So this is just to end. Finally, because we have in this large study multiple baseline replicates of the same donors across different plates, we can calculate the CV in the standard pipeline compared to the CVs that come out of this new pipeline that has this QC and modeling effects taken into account. And you can see that the CVs are pretty much improved. So to conclude, well, we follow all these steps that aim to revisit the way these data are being analyzed. We are working towards an R package and possibly a web-based tool coming soon to put this together. So I acknowledge the Center of Human Immunology at the NIH. This is my contact info, and I'll be happy to take any questions. Thanks.
Getting beyond the improved ability to analyze the data, do you have any results yet showing that the improved modeling leads to novel discovery? Any nice anecdotes yet?
No, it would be great. Not yet. Yeah, I'm still in the phase of getting the method out, testing it, and essentially, so it's been developed on this very large data set, and I'm now validating it on an independent data set. When the method is out, then the interesting part is, yeah, getting to new biology. Not quite there yet. Thank you.